Hello, this is our presentation on Mendelian inheritance in Brassica rapa.
My name's Rebecca.
My name is Alexis.
My name is Luam.
And I'm David.
Next slide. So this presentation will be split into 4 parts. It's going to start with the introduction, just going into the genetics fundamentals, our hypothesis, and more background context. Next, we'll dive into materials and methods and how we conducted our experiment over the course of the semester. And then we're going to go into our results and then wrap up with our conclusion, just explaining our findings and the significance behind that. Next slide. So to start off, major breakthroughs in learning about our inheritance pattern started with Gregor Mendel, who established the 3 fundamentals, which is the law of segregation, confirming that we inherit 1 allele from each parent, the law of independent assortment, that the inheritance of 1 trait does not rely on the inheritance of another, and the law of dominance, an individual with 2 different alleles will express the dominant trait, and that'll be the one to show phenotypically. For our experiment, we used fast plants, and we use them because they have a fast life cycle with no seed dormancy. Many offsprings are produced, they're small in size, and they're easy to grow in a lab setting and relatively inexpensive. Next slide. So we took those laws into consideration when forming our hypothesis for our experiment, which involved crossing 2 parent plants and observing the phenotypic traits of the offspring, which will be F1. So with P1 being homozygous recessive, it had a green stem and yellow-green leaves. And for P2, for the sake of the experiment, we assumed heterozygous, with it having a purple stem and green leaves. And because of this pairing, we expected a phenotypic ratio of the F1 generation to be 1 to 1 to 1 to 1. So it should express each of the 4 traits pretty equally. Next slide.
For materials and methods, we have the potential phenotype of both P1 and P2 plants were predicted as the plant matured in lab. The first appearance of flowers, the P1 and P2 plants were pollinated with a pipe cleaner to produce seeds. Once the seeds were fully matured, they were dried, harvested, and placed onto gel plates to grow. Genomic DNA was then isolated using the Zemo plant seed DNA mini prep kit, and 2 to 3 leaves from both P1 and P2 plants, as well as 1 full seedling of the F1 generation. Small samples of the isolated DNA were then added to a nanodrop 1C UV vis spectral photometer to attain DNA concentration and purity. Next slide, please. The isolated DNA was then used to perform a PCR reaction to amplify the D9B-RAPA S1 locus. Once amplified, the DNA was added into the wells of an agarose gel plate and viewed under a UV trans illuminator to then infer plant genotypes. The purified genomic DNA was then used to run a new PCR to amplify the PARC14 locus. Once amplified, the DNA was combined with the EcoR1 enzyme to cut the DNA at a specific restriction site. A gel plate was then ran and viewed under a transilluminator to determine the PARC14 genotypes and confirm F1 genotype inheritance.
For our results, we analyzed 70 F1 seedlings for stem and leaf color to observe its inheritance. Table 1 shows the observed phenotypes. The majority, 56 out of the 70, had purple stems and dark green leaves. Much smaller numbers appeared in the other categories, with 5 of them having purple stems and yellow-green leaves. 6 of them had non-purple stems and dark green leaves, and only 3 had non-purple stems and yellow-green leaves. Based on a cross between a homozygous recessive parent and a heterozygous dominant parent, we expected a 1-1-1-1 phenotypic ratio in the F1 generation. However, because our phenotypic groups combine multiple genotypes, the expected values were not perfectly even. In table 2, next slide, please. The adjusted expected counts are about 26 seedlings each for the 2 dominant phenotypes and about 9 seedlings each for the fully recessive phenotypes. To determine whether our observed data matched our hypothesis, we performed a chi-square analysis. The calculated chi-square value was 55.56 with 3 degrees of freedom, resulting in a p-value less than 0.001. This is statistically significant and led us to reject the hypothesis, our hypothesis.
For table 3, this is our nanodrop 1 spectrophotometer quantification results of our 3 template DNAs. They quantified the concentration as well as the purification ratios for our 3 template DNAs.
Figure 4 shows the gel plate of the PCR amplified D9B RAPA S1 allele. Starting on the right, we have the 100 base pair ladder. Next to it is P1, then we have P2, and finally all the way to the farthest left is the F1. Next slide, please. Figure 5 shows a gel plate as well. This plate is the PCR amplified undigested and digested EcoR1 Park14 allele. Starting at the left, we have the 100 base pair ladder. Then we have the digested P1, digested P2, and digested F1. And finally, undigested P1, undigested P2, and undigested F1.
As far as our conclusions go, our phenotype ratios deviated strongly from Mendelian expectations. Because the p-value was so low, the deviation was extremely unlikely due to random chance. The dominant phenotypes overrepresented. This was likely due to indistinguishable heterozygotes or human classification error. Our molecular results contradicted the phenotypic deviations, showing that the inheritance at both loci followed expected Mendelian patterns genetically. For our molecular analyses, the D9B RAPA S1 VNTR and Park14 SMP genotyping confirmed the P1, P2, and inheritance pattern in F1. Genotyping aligns with the prior studies showing VNTR-based polymorphism was reliable for parentage confirmation in Brassica species. Our initial Park14 RLP analysis did not produce the expected digest fragments due to incomplete or failed restriction digests. As far as our future improvements, we can look into genotyping all F1 individuals as well as accurate enzyme handling to get a successful restriction digest. We can also increase the sample size for better likelihood of better results.